Awesome. All right. Well, welcome to the Craniotomy podcast, where we peer into the minds of those on the front lines of neuromodulation and neuropsychiatry. I'm Dr. David Darrow, a neurosurgeon here with Dr. Alex Herman, a psychiatrist, and we are the co-principal investigators of the Herman Darrow Lab at the University of Minnesota. Thank you for joining us for yet another very exciting episode, because today we have Dr. Drs Voytek. After completing his PhD at Berkeley, Dr. Voytek joined Uber to help them kick off their data science team and all their efforts when it was very small. He came back to the dark side and started a lab at UCSD, where he continues to bring a very fresh perspective to how we approach and understand time series analysis of the brain, and the brain waves across human disease and the networks that compose them. He has had a number of very high-profile papers, and we are extremely grateful that he could join us today.
Yes, thank you very much for joining us.
Thank you. I think it's funny that you refer to academia as the dark side.
That's true.
A slightly different perspective.
That's right.
Yeah.
I think you have a very interesting background, Brad, and if you wouldn't mind, we'd love to hear about sort of your arc with neuroscience and how your perspective on that.
Oh, that could be a very long story. It could take up the entire rest of this podcast just talking about that arc, right? I actually started my undergraduate wanting to be an astrophysicist. So I growing up, my dad jokes that like when I was 10, he's like, well, what do you want to be when you grow up? And I'm like, an astrophysicist. He's like, I don't even know what that is. I was fascinated by astronomy in general, and then learned about physics and cosmology. And as a teenager, I read pretty much any pop sci book on the topic that I could. Hawking became like a pretty big influence of the way that I thought about stuff. Got to undergrad, joined a lab that did ultra low temperature physics, and felt incredibly lost and didn't like it. So it's like this years long build up in my mind. And I finally got to undergrad, and I finally got in a lab, and I'm like, I'm doing it. I'm doing science. And I was like, oh no, this isn't what I want to do. And it took a long time for me to actually accept that. During that time, I went to undergraduate at the University of Southern California in Los Angeles. And I actually grew up in San Diego. I would have loved to come back here, but you know how academics work. Like I never expected I'd be able to come back here. But I spent a lot of time driving back and forth between undergraduate at USC and then back home in San Diego. I grew up with my step-grandparents because my step-grandfather, just before I left for college, had been diagnosed with Parkinson's. And so I spent a lot of time driving back to San Diego taking care of him. And so I was going through this kind of personal crisis of faith in terms of what I wanted to do with my life. And while simultaneously, watching this person whom I respected and loved, go through this pretty terrible, rapid degeneration. And so out of curiosity, I took a intro to psych class with a friend, and as they said, went over to the dark side of the soft sciences. I ended up going over to psychology. USC had a neuroscience or neurobiology major at the time, but I didn't like the cell biology side. A lot of my friends were pre-med, and that didn't interest me. And so I accidentally cobbled together my own cognitive science degree without knowing what cognitive science was. So I had taken all that math and physics and stuff like that from my first 2 years as an undergrad in physics, and then took the psychology and social sciences kinds of courses and finished out my degree. But a lot of my friends were also computer science majors, and they went in, this is the late 90s, .com boom, and they went and got some really good jobs. And so I said, well, I don't know what I'm going to do with a psychology degree. So just in case, I'm going to hedge my bets, and I'll take a couple of programming classes. And I took a couple of programming classes, really enjoyed that aspect of things. And basically, as part of that process of the difficult couple of years that I'm washing over, I went to register for classes my third year of undergraduate, and they said you can't because you're not a student, you're not enrolled anymore because you've been on academic probation for too long. And so that's when I had the like, moment in life. And I begged and pleaded for one more semester to get my act together. But I'd lost all my scholarships at a really expensive private university, and I don't come from a being able to afford an expensive private university family. And so scholarships were the only way I could stay there. And so an admin undergraduate administrator took pity on me, really helped me out, like one-on-one, a lot of attention. And I got undergraduate work study. So you can work on campus in order to pay for your tuition and pay a little bit of money too. And so I could have worked in, the dorm cafeterias or something like that. But one of the options was you could also work in a lab. And so I ended up getting a job in a lab with Professor Adrian Rain. He's now at Penn. He does the biosocial basis of violence. And he had decades of data that they had in text files that they collected behavioral data from their participants. And my job as the first, grunt undergraduate first job was to open those text files, hundreds, if not thousands of them, copy the data out of them and paste them into an Excel spreadsheet. So that was my job, right? Is like, take this data out of these old files so we can analyze it in Excel. But because I had just taken this very basic intro to C++ class and the data were semi-structured, I wrote code. And so they're like, you've got 2 or 3 weeks it's going to take you to do this, so whenever you finish, you're finished. I came back like the next day and I'm like, all done. And the way I tell it, I may as well have performed a magic trick. They're like, you can't possibly be finished. And I was like, well, I just wrote some code to do it. And they're like, what does that mean? And I was like, oh, wow. So this rudimentary level of programming knowledge, intro to C++ level of knowledge, which was not anything that could get me a job in software development, like my friends who were computer science majors ended up getting, was a breakthrough in this psychology neuroscience research lab. Mm-hmm. And that's what really started, that's where that whole process started coming together for me, of the technical skills that I've picked up, as rudimentary as they are, have strong applicability in these other areas. And that really ended up shaping my career path going forward pretty substantially. So that's the genesis of the entire thing for me.
No, that's absolutely fascinating. And I think what's interesting is that all three of us went to university to do physics. And so it's a really interesting to hear about your arc, because I think we all have sort of similar, a similar mindset. And I think, when you're in a field and they're always pitching it, but the idea that, just the way that you think about physics and the kind of people that it attracts, brings a sort of mindset that, has the potential to, go down a lot of different paths. And I think people, it's sort of the new liberal arts degree in some sense.
Yeah. I would say I was equally influenced by the physics and math side, the programming side, and the philosophy side, right? Like I took a bunch of, philosophy of mind class, and I have a friend who's a philosopher, and sort of with him independent study on epistemology and logic and stuff like that. And all three of those are strikingly similar in the sense of, how do you take really complex real world phenomena and distill them down into a set of rules that you can reproduce, right? And so, philosophy and logic, that's the basis of it, right? Is how do you determine the truth of a statement? That's a very fuzzy thing, but that's what epistemology and metaphysics is centered around, right? Physics, it's okay. You've got these complex interacting systems. Are there a set of fundamental rules that we can use to try and describe this apparently complex and unsolvable problems? And in programming, it's codifying that, writing those rules down and making them into something that is reproducible that you can do every single time. Oddly, I haven't actually thought about how similar the training in those three for me were, but they really did. They're all very similar, just coming at it from attacking it from different sides, right? One is what are the fundamental rules? One is one of the fundamental math, and then one is the implementation side the software development side.
That is a fantastic segue, I think, because I think what's so interesting about your work and what segues from this discussion is essentially how there have been sort of many different approaches towards trying to dissect the electrophysiology of the brain. And basically the role that oscillations of the brain play in sort of circuit level behavior and behavior in general.
Yeah, I was drawn to them, the oscillations. My former postdoc, Natalie Schwarnikov, put it pretty bluntly in a way that I like, which is they're just beautiful. You look at the data and you see these signals and you're like, what? Why is that pattern there? Right? What is it doing? It's got to be there for a reason, right? You record from anything's brain, and you're gonna see these rhythmic fluctuations in voltage sometimes. And historically, right, it's the first signal that was recorded from humans. Hans Berger described the alpha wave in the visual cortex, right? It's this predominant rhythm. People on the podcast, it's 8 to 12 hertz oscillation in the visual cortex. That's predominant in people, humans, when they're sort of at rest. And you just want to know, is it doing something? Why is that there? What does that mean? And there's tens of thousands of papers that have been published over the 100 years, nearly, since their discovery, trying to figure out, are these oscillations in voltage of brain data doing something? And I was really drawn to that. I found that to be, I didn't want to work with animals. I didn't want to, I just couldn't, no ethical reasons. It was personal constitution. I just had a hard time working in animal labs, whereas, people I can talk to and get along with, and we could do our thing. So it's one of the more predominant signals you can record also from humans. And so that was what drew me to them. And that's what got me my job talk here at UC San Diego when I was interviewing, was talking about, the potential functional role of oscillations. And it's really funny that, I joked when I gave my tenure talk a couple years later that, the reason you all hired me, I'm not even confident about this anymore. So let's dig a little bit more. I'm no longer even certain what is meant when the field says neural oscillations now. Talk yourselves out of understanding what an oscillation is.
Don't knock us down too hard, Brad. We're just starting the lab here, okay?
I think that's where the excitement is, right? It's like, okay, if we're going to say that these are doing something, right, have a functional role in perception and cognition, what does it mean that I don't see them all the time?
Right.
They're not always there.
Right.
If you do brain surgeries. I don't know if you do implantations in medial temporal lobe. If you do recordings from medial temporal lobe, you put an electrode into a rat's brain in the temporal lobe, in the medial temporal lobe hippocampus, and you see theta. It is theta. That's all it is. It is this massive 8-ish hertz in rodents rhythm of theta. It's huge. The brain is dominated by it. You put an electrode into a person's medial temporal lobe, into the hippocampus, kind of, maybe if you squint sometimes, you can see it, right? So if we're going to say theta is the Nobel Prize winning mechanism by which rodents learn, how to navigate in an environment through theta phase precession, what does it mean that we don't see it in humans that often?
Right.
And so that's what really started the route that we went down, is what if we're going to say realistically that oscillations do something, what does it mean if they're not there? Are they really not there, or is it a limitation of our measurement ability? And so that's what we've really been focusing on these last couple of years, is, okay, if we're going to be talking about these things, honestly and truthfully, we need to make sure that we're measuring them correctly. And, it's been an incredibly fun research path, for us, for the last 5 or so years that we've been doing this.
So can you expand on that a little bit? Because, we actually want to get back to the 1 over F aperiodic activity stuff you've been working on too. But since we're on the topic of oscillations, where is that taking you? I mean, what is your thinking now about band limited phenomenon? And
Yeah, I wish I had a clear answer. I don't know. I think that I'm really stuck on it because I'm now struggling to say, okay, well, the evidence is such that it is pretty clear that ongoing oscillations can bias perception. People have done this in visual cortex in animals and in humans. You could causally manipulate oscillations through neurostimulation and get the hypothesized predicted perceptual and behavioral effects. You can drive rhythms through optogenetics. Laura Colgan, I think, has done some really cool work here, right? There's some really cool like causal manipulations. Mm-hmm. But then at rest, you don't always see them. Mm-hmm. And so they do something, but they're not the only thing doing the something, right? To like be very vague about it. And so I think that's an exciting opportunity then to say, okay, well, what does it mean then if they are there versus not there? And I guess the thing I get stuck on is, you're both physicians. I joke with my students that what if the electrocardiogram, just nobody really noticed it. The discovery of the electrocardiogram in like 1919, also a Nobel Prize winning discovery. The scientists at the time were able to sort of reverse engineer based upon the shape of the QRS complex and the T wave, the cardiac physiology, right? You're like, okay, here's what the P complex means, here's what the QRS complex means, and here's what the T wave means, right? You can reverse engineer based upon the placement of the electrodes and the leads, and what was known about the cardiac physiology at the time, what those cells might be doing in order to give rise to this characteristic stereotyped wave, right? And I often joke with my students, I'm like, what if they didn't figure that out? And, all we had was the ability to analyze it. Would we be doing like Fourier transforms and filtering of the ECG and talking about like complex nested oscillatory dynamics in the heart? Because if we do the algorithms, if we actually analyze them the way that we analyze neural data, you could find these like really fascinating kinds of effects. But that's not the right way to describe the system, because we know this one already. We know the answer already, and it's relatively simple. Certainly much more simple than, several tens of millions of microcolumns with different orientations and different, neuron densities and cell types and neurotransmitters, right? Certainly far less complex than that. But like, I often wonder if we would be talking about, these things if we just hadn't discovered that in ECG. And so I think about what information can we extract then from our field potential recordings. It is a fact that the visual cortex tends to oscillate between 8 and 12 hertz. And those oscillations in the alpha band have a kind of stereotyped waveform. They sort of manifest as like these sort of deep, sharp V shapes. In the somatosensory cortex, you also have an oscillation at a similar frequency in the alpha range, except it has a different waveform. It's got the mu rhythm, so-called, because it sort of looks like an M-ish shape, right? The hippocampal theta in the rodents, and when it does manifest in the humans, has more of like a sawtooth kind of appearance. Same with the beta oscillation in the motor cortex in humans, right? So there are stereotyped features of these rhythms, and they differ by where in the brain they manifest. And so I wonder if those waveforms are telling us something, right? If we can unpack that a little bit more and sort of think about it from like the ECG standpoint of what can we infer about the physiology of these columns based upon the fact that there are these regional differences in the frequency, the period, the waveform shape, and so on. And so that's kind of where we're getting at is I have an opinion, but I don't have an answer about what these oscillations might be doing, right? The best we can do is say, I am not confident about the historical literature because I think we've been bandpass filtering our data, and everything that you bandpass filter looks like an oscillation, even if there's not an oscillation there. And so I think we've been mixing up real oscillation data with, as you referred to earlier, the aperiodic, non-oscillatory. And so, okay, that's maybe some of the confusion in literature about like maybe visual cortical alpha does gating, and maybe it does rhythmic inhibition, and maybe it does working memory and X, Y, and Z. Maybe some of that muddiness comes from the insufficiency of the filtering approaches. And so let's get a little bit more close to the data and, just see what the data can tell us. So I'm trying to be as agnostic as possible because I want to let the data speak as best as possible. And that's kind of, like I said, where we're at these days.
So you've sort of been pursuing these two approaches to decomposing electrophysiological signals in parallel, one of which is identifying potentially non-sinusoidal signals, like you've been saying. And the other is, it's obviously related, looking to see if there's a change in the slope of the power spectrum fall off, this aperiodic activity. So it's almost as if, and I was thinking about this when you were talking about the ECG example. We've been using the wrong basis to decompose these signals, right? A Fourier transform, it works, but it doesn't mean it's actually the basis that reflects the physiology, right? And that you're coming up with a new, you're attempting to construct a new basis to decompose these signals that actually maps onto the underlying physiology. Yeah,
and I have to say, I think Steph Jones at Brown University has been doing really cool work on the non-sinusoidal stuff too. She's taking a different tactic, which I love. Right, I wish we had done it. She created HNN, I think she calls it, the Human Neocortical Neosolver, which is saying, okay, let's try and simulate cortical circuits and see what kinds of waveform oscillations can manifest in simulation.
Right. What even comes out? Yeah.
What even comes out, right? And so the idea there, I guess, would be, and I haven't talked to her for a while about it, so I don't want to put any words in her mouth or anything like that. But I think the idea there would be, okay, given the fundamental limits to neurons, right? Neurons exist in a finite world. They have finite shape. They have refractory periods. They have specific kind. They're not oriented randomly, right? You've got the pyramidal cells that have a nice orientation. Interneurons might be oriented a little bit more randomly, but there are rules still to neuroanatomy and neurophysiology. If we simulate those rules and tweak the parameters to within physiological bounds, right? Like a neuron action potential can only be so high in amplitude, right? It can't be 10 megavolts, right? They exist within these bounds, right? And so let's sort of sweep through all of these physiological bounds and see, are there stereotyped patterns that emerge, right? And so I think a really cool, I don't know if it'll work, it might be totally mathematically intractable or degenerate, but it would be really interesting if in that simulation you could say, actually, it looks like physiologically there's only these 53 patterns that can emerge, right? That's it. There's only these 53, right? Anything else is non-physiological within the bounds. And then you have a dictionary of basis functions that are physiologically determined, right? You can either have an M shape or a sawtooth or a reverse sawtooth, a deep V. Maybe there's only, I'm just making up numbers. It's not actually 53, right? Maybe it is, but that would be but that would be another way of going about it potentially, right? Is alterations in these waveforms might tell you something about the underlying circuit. I don't know. We can't know until we have good ways of measuring them.
And I think that's, part of the struggle that, there's a lot of tension where we're sitting, where, the reason that I went into neurosurgery and the reason that Alex went into psychiatry was to sort of look at these sort of biological underpinnings of psychiatric disease. And, that assumes that we know anything about the underlying biology. And, it's a constant, I think Alex and I look at each other constantly because we see so much variation in the recordings from people's brains that it's always you get this feeling is this just, an epiphenomenon of sort of a stable state that optimizes information transfer in these circuits, and then there's a lot of variability because the manifold is just enormous, or
what? So many dimensions, right? Not infinite, but very massive,
right?
And so it can take a ton of different forms,
right?
Which is why I think the oscillations are so beautiful, that you have this level of complexity, and yet you get these stereotyped features that emerge.
Exactly. So it begs the question, which is, separate from, what you're trying, what we were talking about before, but something that you're kind of working on, which is essentially of the sort of question, how personal do we need to get in order to link this with biology that plays a role in, mental illness or development, or, are we able to gather enough information, as a group to understand an individual so that we can make changes that make sense to us in a systematic way so we can improve people's lives. I think that's what we're constantly worried and, focused on.
Yeah, I mean, coming from a cognitive science department, right? I feel like I've been infected by the cognitive science thinking, right, in a good way, which is the 1990s, right, were the decade of the brain. Huge expansion of the NIH, and trying to understand the biophysical basis, the neurobiological basis of mental health and mental illness.
Yep.
And huge strides were made in genetics and in neuroscience and neuroimaging, and yet we still don't have diagnostic. The diagnoses are still done by physicians meeting with the patients and saying they have these set of symptoms that have caused personal difficulties in their lives in the last 6 months or whatever, right? And of course that's the DSM based diagnoses, and there's sort of the move by the NIH, I guess, to try and go to the RDoc, right? But we still don't have that biomarker, right? There's a lot of putative biomarkers out there, but very rarely, for very few diseases anyway, do we have neurobiomarkers. And so on the cognitive science side, you say, well, then maybe the neuroscience is not enough, at least at the level that we can measure it, right? You think about mental illness is certainly affected by genetics, but it's also affected by the environment. It's the gene-environment interaction, right? And so two people with the exact same genetics who are raised in two different environments can have totally different outcomes. And so, like on the cognitive science side, you think about what is the sociological context in which people are embedded, right? We live in a particular time in life where we have now gone through 2 years of quarantine lockdown, right? That will certainly have differential effects on mental health that are inexplicable from genetics alone, right? Because it is now 2 years of this lived experience for people, and those experiences are different depending upon the country of origin, socioeconomic status, and so on, right? And those will then manifest also neurobiologically, right? Those 2 years of lockdown and any sort of mental health issues that arose from it or not are going to have longer term influences on brain development and aging and stuff like that. And so I struggle with it because it's so complex, right? And the physicist in me wants to say, well, okay, yeah, but maybe we can't solve the 3-body problem or the n-body problem of the motion of planets, but F equals ma still, right? We still have some fundamental laws, and that would be amazing if that was the case, right? Because if you find that, you have the opportunity to dramatically improve the quality of lives of many, many people.
I mean, I think that's exactly what you said. The F equals ma in medicine is, the social determinants of health. That it's almost, paradoxically, and comically the opposite, but that is absolutely the basic foundation of medicine and health is, society matters, environment matters, right?
Yeah, the number of adverse childhood events is all that you need to really know.
That's right. That's F equals ma. The other part is, I think where nobody knows really what to do. I mean, everybody has great ideas. Right. You have fantastic ideas about how we can start looking at these biomarkers and get more separability and in a way that makes sense biologically from a very fundamental basis. But it's a strain to be confident about, how much effort, everybody's putting into medicine when, still F equals ma.
Right. Yeah.
I'm going to make a leap here, which might be a little bit of a stretch, but bear with me. So I've been reflecting on your recent eLife paper, which I just read prior to this, and one of the things I think was so interesting about what you tried to do there was dissociate the encoding of sort of environmental statistics and the sensory task in, for instance, the aperiodic signal versus the oscillatory signals from sort of endogenous rhythms or cognitive processes. And so this was coming to mind as you were talking about, we're all embedded in the sociological context, right? Because on some level, when we're recording brain activity, we're not just recording things that are being generated, within the brain. We're recording the results of our, the way that the brain is actually responding to or even representing the statistics of our environment.
Right.
And so just to try to make a link to the, actual scientific work you've done, I'm just curious about, like, and I don't remember from that paper, how strong were the relative components of those signals, like the representation of the sensory statistics versus other things that you detected in the brain signals.
So this is the Washke eLife paper?
Yes. Yeah.
Yeah. Yeah, I don't remember how strong like the different components were. Well, so one thing that we found pretty fascinating was so for a little bit of context here, for people listening, right? One of the I'll talk about the study specifically, but like the bigger picture underlying it, right, was if you present somebody with white noise, either visually, so like static on a TV screen, or sound, right? I can't do it, of course, but it's something like, right, kind of white noise sound.
Really good. Yeah.
Versus more structured noise, which I absolutely can't do, which would be what are called like pink noise or brown noise, where it's random, but it's random in a different way, where there is some structure to the randomness. So white noise means that what's happening right now is totally independent from what's about to happen. That's what static is. Every piece of the signal is totally independent. Whereas you can have other kinds of noise, but it's not independent in time necessarily, right? So it's still random, but there's some structure imposed where you can predict a little bit what happens at time t+1 from what happened at time t. And that's sort of what's the analogy people use, like avalanches or something like that, right? Where the snow falls randomly, but it can build up and accumulate slowly, and then you get an avalanche, and then it sort of mounds back up and then falls, right? It's still random, but there's structure in that randomness. And the brain activity, this aperiodic signal, this non-oscillatory signal in the brain that we can measure, seems to track the statistics. So if we present white noise, the brain activity looks more like white noise. It's not white noise, but it looks more like closer to white noise than if you present more structured randomness. And the idea there actually is like our bigger term vision is we've been focusing in the lab about how do we quantify our signals and what information can we extract out of the brain data from the shape of the oscillations, are they non-sinusoidal, V-shaped, sawtooth shaped, to this aperiodic activity. And so I guess I should spend a couple of seconds explaining this aperiodic activity. If you look at brain signal data, it looks kind of random. If you look at how does voltage vary over time in the brain signal. And that randomness has statistics. It's not like white noise random. It's more like the brown noise avalanche is random. And sometimes you also then have these waves on top of that randomness, or in addition to that randomness, or maybe as a consequence of that randomness. We don't know. But that randomness isn't random. That randomness comes from biology. It's being driven by large groups of neurons that are acting together, either in synchrony or out of synchrony or to varying degrees of synchrony. And so if you can quantify the data just right, you can see what kind of randomness is it. What's the shape of that randomness, right? How structured, unstructured, or semi-structured, or fully structured is it? We could quantify that. And the idea is the direction that we're starting to go, at least one branch in the lab, is what if we take the same idea of quantifying our data in like as rich of a rich way possible. So rather than saying what is the frequency of this oscillation, and how big is the oscillation? What's the amplitude or power of that oscillation? Which is what we traditionally do. We could say on this 100, if it's a 10 hertz oscillation, each cycle of the oscillation, for people who might remember their like trigonometry, right? A sine wave, it's a regular oscillation. If it's 10 hertz, that means it happens 10 times per second. So one cycle of this oscillation happens in 100 milliseconds. What is the amplitude of the peak? And what is the depth of the trough? And is the peak smooth or sharp? How sharp is it? Is the trough smooth or sharp? It's not exactly 100 milliseconds from every peak to every peak. What's the exact period from this cycle to the next cycle? And like, how quickly does the oscillation rise in time, and how long does it take to decay, right? And so we can measure every individual cycle and leverage the variance in our data. So instead of just averaging across and say, look, it's an oscillation that's on average 10 hertz, we could say on every individual cycle, there's a little bit of variance. Let's take advantage of that variance and see if that variance on a cycle by cycle basis relates to the experience of the person, right? And we're starting to do that now also with the stimuli that we present, right? So if I grew up in my PhD days studying working memory and attention. And the way that you study working memory and attention in people is you stick them in a dark room and you flash a bunch of squares on the screen and you ask them to remember, 1, 2, 3, or 4 squares, and they flash on the screen, they go away, and then you present the same number of squares in the same spot, and you ask the person, did one of the squares change color, yes or no? And did they get it right or did they get it wrong? And that's how you study working memory, for example. This is how I did it. I published papers on this. But that's not capturing, that's really contrived. That's not what memory is like in the real world, right? But coming neuroscience, cognitive neuroscience, having grown out of cognitive psychology, which partially grew out of behaviorism, you're trying to say, let's ignore, let's try and smooth out everything else and control all of these other features and just study memory. How can we distill memory down to like the simplest component with as few distractions as possible, which is where those kinds of studies came from. But, what if I kind of remember the color of the square? I don't remember if it changed or not, but I can tell you what color it probably was. It was greenish, right? So it's not a binary, do you remember it or not remember it, right? It's I kind of remember the color. How well you do is a function of how many things you have to remember. But what if, you have to remember 7 things, but they're all vaguely bluish? Then is it really 7 different things you're trying to remember, or is it kind of chunking, they call it, together into like one? And so what we're trying to do is we are now quantifying as much about the stimuli we present to people as well on a trial by trial basis. How many things did we present? How spatially clustered were they? Are they really tightly spatially clustered, or are they more spread apart? How perceptually color clustered are they? Are they like on opposing ends of the color spectrum, so you have to remember very distinct? Or are they kind of close together? How far apart are they? What's the size of them, right? And so we can actually start to say with the data analysis approaches that we now have access to, not just look, here are the parts of the brain that are, where the activity scales with working memory load, the number of things you have to remember, but you could say, which parts of the brain scale most with load? Which scale most with perceptual dispersion of the colors? Which ones scale the most with spatial dispersion, right? You could start to sort of build up these more sophisticated ways of mapping experience to brain data rather than controlling for everything. We don't have to control for everything now. We can be a little bit more expansive with more modern data science and data analysis approaches. When I teach my undergraduate data science classes, I'm in a social sciences department. And so in the social sciences, people who do ethnography and social sciences will talk about, they do qualitative data analysis, right? They'll go out and they'll have interviews with people and they say, what is the impact that this legislative change is having on your experience, right? This change in welfare policy for this state that was enacted at this point, how did that change your ability to pay for food and help your children get them to school, the work hours, right? Those are conversations that they have, and they record those conversations, and then they sort of read through them to try and distill out some common themes, right? And that's qualitative data versus what we're talking about, which is qualitative data. And I think one of the amazing things about sort of the modern data science approach is that the distinction between qualitative data and quantitative data is blurring, right? You can quantify the qualitative better now. Right? We could take those structured interviews that are conversations, and you can run them through natural language processing algorithms to try and distill out themes, right? And so just the same, we could take the visual stimuli or auditory stimuli we present with people, and we can quantify them more richly, right? We could say, instead of having to smooth out and just like control for everything, let's actually start letting more variability in, as long as we're tracking that variability to the best of our ability. And so that's sort of one of the themes that we're running with in the lab. And hopefully then that'll let us get to a little bit more, if you have enough data, which is going back to what David was saying earlier, if you have enough data, not just from people, but of what the people are experiencing, then you might be able to find better relationships between these neural features and genetics and so on to behavior, cognition, and maybe disease, right?
Mhm. Yeah, so that's fascinating. So you have this new tool set for sort of more physiologically, let's say, decomposing these signals, even on a cycle by cycle basis, right? And at the same time, you're introducing more variability into your experiments in a way that's like where you're tracking it. Obviously, you're able to account for it. And so I'm wondering if you see kind of putting those two approaches together as enabling you to reveal sort of more individual differences that are actually meaningful. And because, ultimately, when we talk about wanting to identify biomarkers, and one of the things I remember, like right pre-COVID, was there was a lot of chatter from people about using your spectral exponent stuff to, find biomarkers for deep brain stimulation. And I think I heard a talk recently where that came up again, COVID black hole or whatever. But so I'm just sort of curious, like, are you more optimistic now that we'll actually, given these new tools that you're developing, that we'll be able to really pull out, what makes some, the individual's brain, essentially contribution to their own brain signals as opposed to the environment or the task or whatever it is?
That's a little bit of an unfair question because I'm eternally optimistic to a fault that annoys people. I guess it's like my coping mechanism in general. But no, I am optimistic. I am excited about the future of neuroscience, but I could say that always, right? I think there's always new exciting developments. That's what makes science so wonderful. That's why I do this job, right? Is there's always something new to learn, and we just learn from mistakes and get better, hopefully. But I am optimistic that there will be more we can do. Now, I have to say that this isn't, a uniquely me idea. I grew up in an environment academically at UC Berkeley in, the 2000, late aughts, early tens, where people were starting to do this kind of like data-driven approaches a little bit more. More famously, Jack Gallant, UC Berkeley, who was doing single unit physiology for a long time, started getting into fMRI in humans. And I remember he had an experiment. Alex Huth is one of the first author. He's now at UT Austin. Had done this experiment, which was basically going at the classic, I think it was 1999 Nature paper from Nancy Canwisher, looking at that sort of identified fusiform face and perihippocampal place areas using fMRI. Right. So the idea for those who aren't familiar is that you can have somebody in a brain scanner and you could show them simultaneously a picture of a house and a face overlaid on top of each other. So you sort of like semi-transparent, like overlaid, and you just ask people, pay attention to either the houses or the faces. And when you do that, and you look at the fMRI brain signal, you see that there's a certain brain area that we refer to as the fusiform face area that is especially active when they're paying attention to the faces. And then another one called the perihippocampal place area that's especially active when they're looking at the houses, the places. And so they're seeing the exact same thing because they're overlaid, but it's depending on what you're paying attention to, you get these differential activations. And so people are like, aha, you've got this brain mechanism, a brain region dedicated to face processing, and another one that's dedicated to like place processing. And Jack Gallant gave this talk, and he's like, you can't say that. All you can do is say that the fusiform face area is more face than house, and that the perihippocampal place area is more house than face. But maybe it's a toothbrush area, or maybe it's a rainbow area, or a skateboard area. You don't know because you didn't test. You only tested two kinds of stimuli, right? And so Jack Gallant and Alex Huth came along with a couple of other collaborators and stuff, and they just presented movie clips to people. And they annotated every frame of that movie clip with what was in the frame at that moment. And so, it's like maybe a woman riding on horseback, wearing a cowboy hat. And so you'd have horse and woman and hat, right? But they also then did the sort of like natural language processing on the features to look at semantic similarity, right? Where a woman and a horse are both animate mammal living beings. And so they're closer, more similar to each other than like a hat is to a horse, right? And they then did this, annotated all these things, showed, I think people were in the scanner a total of like a dozen hours each looking at these things, and then did whole brain regression maps to see what are the brain regions that are responsive to hats or animacy or horses or whatever. right? You get these rich, incredible maps of brain activity in response to all these different things, right? And you're like, look, that's the reality, right? There isn't just a fusiform face and parahippocampal place area, right? Our representation of the world is much richer than that. And it almost certainly varies with individual differences. If you've never seen a horse before, maybe you're going to have a different response to the horse than, somebody who is, dabbles in equestrian, what is it called when you're a horse rider? Like dressage, let's just say.
Dressage, yeah.
Right? You're going to have very different neural responses. And okay, we don't care necessarily about that difference in, if you've never seen a horse versus you're an expert in dressage, right? But you can see maybe where you could go with this, where, okay, let's actually take some things that maybe do matter for mental health and start to account for some of these things, right? A little bit more richly, and see what are the whole brain responses to this rich set of features that we can quantify. I guess my only concern with these approaches in the long run is that I don't know if that's going to get us closer to distilling out the F equals MA, right? Like, we might be able to build AI models that can make these predictions for us, right? Which would be amazing from a diagnostic and clinical standpoint, right? It doesn't matter that you don't know that the active ingredient in your bread mold poultice is penicillin. You just know that if you put it on the wound, you're going to prevent infection. That's great. And that worked for hundreds of years, right? But as clinicians and scientists, you want to figure out, what is the penicillin? What's the active ingredient, right? And so my concern is that these sort of data-heavy approaches will lead us to clinical diagnoses and maybe even some treatments. But as a scientist, it's unsatisfying when you don't know why it's working, or you don't know what it means, right? We want to understand why it doesn't work. And so I'm excited about these data-driven approaches. I love it. I teach this stuff. It's my bread and butter. But I am concerned from a scientific standpoint that we still won't figure out what the rules are. And I guess the one thing I'm not optimistic about, really, my concern is we can't know. That's my fear. That's what keeps me up at night. These systems are too complex, the brain and the societies in which we're embedded, that we maybe can't understand it. Maybe there are no fundamental rules, right?
The brain understanding the brain, right?
Yeah.
I mean, I'm sure people felt that way about the heart at some point too, and so I think
that's true.
You can see the heart pump at least, but yeah.
And we can see the brain function. I mean, there are some rules that are there, but it's hard because it's hard in a lot of ways because from my perspective, very few people touch the brain. We touch a lot of animal brains, but we sort of feel like there's kind of there's sort of an ethical dilemma about touching too many people's brains, which there sort of should be. So there is a real bottleneck, versus fMRI and things like that, which are very filtered data, so to speak. It is even more complicated because we're, trying to understand the system of a system, and it's really much more challenging than I think the DSM originally imagined when they were trying to put a lot of fMRI stuff into the latest version.
Yeah.
I think this point about AI, we might potentially develop AI approaches that actually are very useful in diagnosis. It's so important because people have made some recent success, right? Looking at EEG signals, but ultimately the studies are still well, is Zoloft going to work better for this person, or is Prozac going to work better for that person? And we barely even know how SSRIs work to begin with. And so, yes, of course, there's definitely utility in figuring out who should get what medications. But if we can't get somewhere closer to the F equals MA, we're never going to come up with the next generation of interventions that we understand, and B, that can be, more targeted in terms of the way they're actually designed from the beginning.
Right. It's hard to write a preliminary data section on a grant or on an IRB to do a study when it's we use this algorithm. We don't really understand what's going on here, but we see a little bit of separation. And even though we don't understand the biology, we want to risk either huge, vast amounts of resources or people. It is a tough sell.
Yeah, it's really hard.
And we struggle a lot in our work, because we work with these intracranial recordings, which you have, obviously lots of experience with yourself. And one of the things we commonly encounter is we see, information everywhere, right? No matter sort of any part of the brain we're recording from, all over the brain, we might see information, about a task, a particular task variable. And so then, trying to explain that and to understand it is very challenging. This sort of everything is everywhere problem is, it's a fascinating scientific problem, and I think it actually is related to Jack Allen's work that you were talking about.
Yeah.
But it makes the clinical correlate that much, more even harder to imagine. Like where do we put the DBS electrode? Like if
exactly.
Yeah,
and I guess having grown up in the like box and arrows neurology side of neuroscience, right, where it's like this is the module of the brain that does this, and this is the module that does this, right? And as we get farther and farther along, that comes from the clinical studies of looking at people with brain lesions. And yes, if you have a lesion in inferior frontal gyrus, you are going to have some kind of Broca's aphasia, right? But I guess unless Nita Dronkers came along and said actually it's more like anterior insula maybe, but it's hard to get one without the other, right? And that was a tremendous advancement in our understanding of the neurobiological basis of complex cognition. But as we get better and better recording methodologies, Alex, like you said, you can see everything everywhere. And so yes, there are statistical modules. Yes, if you have a lesion in Broca's area, you're probably going to have Broca's aphasia. But there's also, you can, I'm sure, David, you've done, electrical stimulation mapping. You can stimulate like parts of parietal cortex and you'll suddenly get an aphasia like, stutter briefly. Like, well, it's not anywhere near Wernicke's. Like, and so you look at the maps where people have done these electrical stimulation maps and say, okay, what are the regions where you stimulate and you get Broca's aphasia? And they're clustered around Broca's area, but they're all over the place, right? And so this is where the precision medicine comes in, right? I had a colleague years ago who was really into precision medicine. And he said, look, if I go to the doctor, I don't want the doctor to say, you need to take this proton pump inhibitor and that'll help you out, right? Because studies have shown that this will help people out. He's like, I don't want to know studies have shown it'll help people out. I want to know, does it help 50-something-year-old men of South Asian descent with my genetic background? Does it help in my diet? Does it help them out, right? And that's where you get into the precision medicine stuff, right? Up until, 10 years ago, the NIH was conducting drug studies only on male mice. And it's like not even accounting for, female mice until 10 years ago or something, right? It's ridiculous. And so it's the same kind of thing for the neurosurgery and then for the cognitive stuff. It's everywhere, kind of. And what does that mean in terms of how we think about and how we map brain activity? And we've gone well beyond the box and arrows models of neurology. And now we're into the network models, right? Everything is interconnected with everything else.
Right.
And I don't know what the next level past that is going to be, but it's probably going to be more complicated, more frustrating.
Well, we also very optimistic, despite it being incredibly challenging, and continue to try to. I don't know. We've been pushing on sort of really trying to do thorough N-of-1 experiments with good GLMEs to really try to pull knowledge back together, but be able to simultaneously focus on individuals and take that personalization into account. And I think that's kind of where we found a little bit of a happy medium, at least on in the invasive world, because. I don't know if you've seen some of the work that's being done invasively for, depression from, Nader or Samir, or, Helen or any of these people. But the approach now is really like personalized invasive mapping, which is, it seems to be gaining some traction, but is also very resource intensive, in every way that you can imagine. And is also not for everyone, right? We all have this hope that if we do it in enough people, that we'll kind of understand things enough that we can do it non-invasively, because, we are not here to do surgery on everybody that comes in with depression, right? We're trying to keep people away from, serious treatment refractory depression and suicide. But we're trying to pave a way forward. And linking that to all the other smart people out there that have the ability to sort of push those boundaries is super challenging, just given the way grants are funded and how it's all siloed.
I'd written years ago, like 7 years ago now, a review paper, and I ended it by saying, look, we do for epilepsy, you do invasive recordings in epilepsy. The reason we have these intracranial data usually is to identify the epileptogenic zone, right? You can't do it non-invasively with the spatial specificity needed in order to identify where the epilepsy is originating from. And so the patient gets implanted for a week or two with the chronic electrodes to identify the epileptogenic zone, because you could see it clearly in the electrophysiological signals. You could see the ictal spiking, you could see where it's generating. And so I was positing, what if the sort of the strange successes and failures that like the Helen Mayberg approach has had for deep brain stimulation for depression, what if we had that biomarker in the signal where instead of implanting based upon the tractography like she was doing, which worked kind of, right? What if you did a week or two of epilepsy monitoring unit like recording near the implantation zone, and see if we had hypothesized or theorized in this review that maybe there are either oscillatory and or aperiodic markers for depression around the confluence of the different tracks that she's arguing for. Right. And we do a week or two of monitoring and have the patient actually report when they're, because depression states wax and wane as well, right? They're not as rapid as an epileptic event, but over the course of a week or two, you have some days where you feel on, some periods and you're like really, you start to dissociate and feel terrible, right? According to the patient experiences. And what if you could track those and then see, like, are there markers, right? And use that then to guide the implantation either of an invasive deep brain stimulator or hopefully some kind of like targeted non-invasive, TMS or something, right?
Well, you predicted it then, because Eddie Chang and Samir Sheth and Nader, they all have done this, and they are a few patients into their prospective trials. It's
oh really? That's actually ongoing right now?
Oh yeah,
cool.
Yeah,
that's
yeah,
great.
Exactly.
Hopefully they cite you.
Yeah, I think Eddie Chang published his paper. Where was that published, Alex? In
it was
Eddie, so it was probably Nature, JAMA.
Right. It actually was a Nature subjournal.
Yeah. Exactly. Yeah. No, they published theirs, and they did exactly that. So, I mean, I think that's kind of where there's some traction there, and I think for good reason, because we have like reasonable hypotheses that we can test, and you can do some simulation, and you can get some effects that are with a risk profile that's okay. It's pretty darn safe these days. But, it's still surgery and things can go wrong. So it's just one of those things that, we have to really restrict. But the hope, I think, there is not to do, 1,000 patients over the next 10 years amongst 5 centers. It's, to really answer some fundamental questions, if possible, that I think everybody else is hoping for. And I think maybe, hopefully through NIH mechanisms of data sharing and things like that, people will be able to use that data. But I think the hard part is, as analyzing retrospective data is a different sort of thing than doing real hypothesis-driven, work.
Yeah. I mean, a lot of my work actually, the last couple of years especially, has been using just openly available data sets.
Right.
Like we have, I had a real struggle early on, especially getting grant funding. And so I couldn't collect data at the scale that we wanted. And but then kind of came to realize that we didn't need to necessarily, because at this point there is enough open data sets out there that we could start actually pushing at some of the things that interested us, just using the publicly available data sets, right? So I'm a huge advocate of open data sharing, with the caveat that there are many privacy concerns that I'm like, I don't even know how to address, in terms of like identifying, I don't think there's any possible way, if you've got de-identified time series of neural activity, you could identify somebody from that, right? It's just you can't do it, right? So I'm okay with that, but it gets a little bit more dicey when you start including a little bit more personalized information. Yeah, exactly. But Yeah. Anyway, I mean, as always, like I was saying, I'm very optimistic, right? I don't have, I have nothing but joy for this field and where it's going and the opportunities. And, ultimately it comes down to helping people, right? I mean, you're physicians, this is your day in and day out, this is what you do. And Joseph Parvizi tried to convince me to go to med school at Stanford. And I told my wife, I'm going to go to med school. It's going to be fun. It's going to be great. She's like, no, you're not. You're not going to go to med school. Like, don't do that. She's like, you don't have, essentially, you don't have the constitution for it. She's right. But the science work that I do, hopefully, is something that can be leveraged, right? And so, yes, I'm not seeing patients on a daily basis, but hopefully the kind of work that my lab is doing, if not directly, will then influence downstream things that will then influence, patient outcomes, right? And I think that's one of the beauties of this field is that the work that we do is personal. It may not be individually personal, but ultimately it will have downstream effects that should help individual people. That's the hope.
Absolutely. I mean, I think there's no doubt, what you have produced, has changed the way we think about things, definitely. And, how we approach problems in both invasive and non-invasive realms. So absolutely. And there's no doubt tons of people are using your work. So there's no doubt that, what you're doing is changing lives.
Oh, thank you. I hope so. I hope that's true. That's the best.
Absolutely. Absolutely.
And to that end, I want to close by thanking you for publishing your Python toolboxes, publishing papers. It's such a wonderful service to the community.
Yes.
And we use it, for teaching students, everything from, because you have some very basic tools there up to, more complex, sort of non-sinusoidal signal detection stuff. And it's just thank you for doing that. It's
well, thanks. It's great to hear. I mean, that's why we do it, right? We hope people use it.
Yep. And people do.
That's
and
wonderful.
Thank you so much for joining us today, Brad. We really, really appreciate talking to you. I think the listeners are going to love hearing what you have to say.
Yeah, I appreciate it. Hopefully I'll actually get to come out to Minnesota and actually see you guys rather than just virtual, or, if you're coming to SFN in San Diego this year, let me know.
Absolutely.
Yeah, that'd be wonderful. Yeah.
All right, Dave and Alex, thanks guys.
Take care, Brad.